Hi everyone, welcome to our event. This event is brought to you by Datadogs Club, which is a community of people who love data. We have weekly events, and today is one of such events. If you want to find out more about the events we have, there is a link in the description. We actually have quite a few events in our schedule. We have a few workshops, so if you're interested in that, check this out. Then, very important, do not forget to subscribe to our YouTube channel if you haven't done this yet. So there is, this big subscribe button. Click on that. You'll get notified when you do this. You'll get notified about all our future streams. Last but not least, do not forget to join our amazing Slack community, where you can hang out with other data enthusiasts. So there is a link in the description. And then during today's interview, you can ask any question you want. There is a pinned link in the live chat. Ask your questions, and we'll be covering these questions during the interview. So now I will get the questions we prepared for you. Okay, so now the questions are on the left, you're on the right, and we can start.
Okay.
Yeah. Okay, wait, I lost it. Okay. This week, we'll discuss how computational biology and AI intersect to advance cancer research. We have a special guest today, Rileen. Rileen is a senior computational biologist and cancer data scientist who has worked with leading institutions such as MCKSS. You'll need to tell us what they are. I don't know. Then Dana-Farber Cancer Institute and Mount Sinai. His work spans cancer genomics, tumor modeling, integrative multi-omics, machine learning application in translational medicine. He has published first author papers in Nature, Nature Communications, and Cell Reports Methods, and continues to explore AI and ML through hands-on courses and Kaggle competitions. Welcome. So
Thank
that's
you.
a long and interesting bio. And usually the first thing I ask our guests is to tell us more about your career journey. So tell us more about how you arrived at this point of your career. What have you done so far?
Yeah. Yeah. So as you can see, it's a little long. I'll try to make it as short as I can. So yeah. So my background
That's a
is a
good thing, right?
mix. Yeah. Yeah. So my background is a mix of physics, maths, and computer science, eventually leading me to computational biology. And I did my undergraduate studies and my first master's in India. And while I was doing my master's at the Indian Institute of Science, I liked my elective in computer science so much that I decided to do a master's in computer science, which I did from King's College London. And there, an elective in computational biology led me to doing a PhD in computational biology. And I did that in Germany, working with a group of biologists in Jena and computer scientists in Freiburg. And during that, I worked on prediction of alternative splicing. I can explain that in a moment later if there's interest. And after that, I moved into cancer research, doing a postdoc at MSKCC, which is Memorial Sloan Kettering Cancer Center in New York. So I
Okay,
moved from Germany to the US and I've been in the US ever since. So, and I've been in cancer research ever since as well. Did a postdoc at Sloan Kettering. Then I was a staff scientist at Mount Sinai in New York as well. Then I moved to Boston to be with my wife who had moved here for a job. And so then I worked at Brigham and Women's Hospital and Dana-Farber Cancer Institute, which is the Cancer Institute of Harvard. Then I moved to the industry to hope to have some impact on patients. And unfortunately, the company that I joined was shut down by its parent company.
hmm
And so right now I'm in between jobs.
Yeah.
And I've been dabbling in machine learning since the beginning of my PhD, and we can discuss that in more detail later. But in the last couple of years, I've spent more time on upskilling in my own. And as part of that, I did courses from Data Talks Club.
hmm
And so I've done three of your wonderful courses. I started with the machine learning Zoom camp, and then I've done the LLM Zoom camp and the machine learning engineering Zoom camp.
Yeah, that's amazing. I mean, sorry to hear that you're in between jobs, but every time then there is a change. Maybe this change is for good, right? So
Yeah,
I hope
hope so.
you resolve this very quickly and find a job that is even better than the previous one.
Thank you.
You had quite a nice career, very interesting one. So now you live in Boston, right?
Yes.
That's my
Ac
favorite American city.
uh-huh.
It's very
Yeah.
European, at least it seemed to me when I was there.
Yeah, you kind of get a mix of a big city and a small city in some way. Like New York
Yeah,
is undeniably big, but
yeah,
Boston is kind of a mix.
yeah. It was. I felt very nice there. Like I felt like in New York, it's too chaotic. Like it's
Mm-hmm.
all over the top. Like too many things are happening. It's like cognitive overload for me all the time.
Mm-hmm. Yeah.
But Boston is better with this regard. But I was there only for summer. I worked in Boston University.
Mm.
No,
Nice.
cool place. Okay, yeah, you had quite a journey, and yeah, thanks for mentioning the courses. It's yeah, hard. I hope you learned a lot, something you
Yeah,
can use at work
definitely.
too. Um, so you mentioned alternative splicing, right? And you said
Mm-hmm.
we could talk about this. I even took a note,
Yeah.
alternative splicing. So I know what the word alternative means, but
Mm-hmm.
splicing not
Yes,
really.
yeah.
Like, can you tell us more?
Yeah. Yeah, so the only danger is of me talking too long about my research, right? So I try not to. But so splicing originally comes from how you show movies in the cinema hall, you know? So you had these long reels and then they had to be cut and joined together in the right
I
order.
watched Fight Club, right? Have you seen Fight Club?
Yeah, a long time ago.
They
But they show
show it
that?
like,
Yeah.
yeah, like
Yeah.
it's completely random, but like at some point of time in the movie, they're like, okay, this is how it's done. Like you see, like they were talking about, like that this dot that appears
Mm-hmm.
in the movie in the theater.
Mm-hmm.
So like, look, do you see this dot? It means that now it will be,
Okay.
changing
The,
the bundles or how do you call this tapes, right?
so this describe the process.
Yeah, and then he was making pranks also.
Yeah. Yeah, so from that general sense, in the specific sense of biology, what it means is that the human genome is divided into stretches that code for a protein and stretches that don't. So these are called coding and non-coding DNA. And then within the parts that code, it's not necessarily the case that the entire stretch is used for making a protein. Usually, these stretches are divided into, again, coding and non-coding parts, which are called exons and introns. And so, what the machinery which turns genes into proteins has to do, it has to correctly identify the parts which are required for a protein and put them together before a protein is made. Now, if you think from the combinatorial perspective, if you don't always use the same parts, but you use some parts some of the time, then you can have an explosion in the number of possible proteins from the same gene. And so alternative splicing refers to this phenomenon where the splicing machinery can pick and choose different parts from a given gene to make multiple proteins.
Okay. Well, I must admit I didn't understand much, but yesterday actually, so good thing.
Mhm.
Yesterday we had an interview. I had an interview. It was about biotechnology and bioinformatics. So we talked about proteins yesterday too. And well, now I have a little bit of background to understand what you say, but still not enough, right? So
Yes.
it's a little bit complicated. So you say that there are some the genome, we have
Hmm.
coding and non-coding DNAs, and in
Hmm.
order to make proteins, you need, and then I got lost,
Yeah,
I think.
so that is incredibly complicated, but I'm trying to keep it simple by saying that now when you have a portion which is defining a gene, then each gene makes at least one protein. Some
Mm.
genes will make one protein and some can make more than one. And the way that happens is through alternative splicing because this gene is divided into different parts, some of which code for a protein. If you take all of them together, then you have what is called as the longest protein that you can make from this gene. That's like the principal form, if you will. And then if you leave anything out, you will get a different isoform, a different variant of the
Mm-hmm.
protein. And so the more the parts there are, and the more the ways in which they are combinatorially chosen, you can have dozens or even hundreds of proteins from the same gene.
Mm-hmm.
And it's been shown that in Drosophila, in a fly, there are even genes which have thousands of isoforms.
Mm-hmm.
So this
And
can give you a lot of related proteins with different specialized functions, which is a part of how we function as humans. And so the machine learning task was really that given a lot of exons, which are conserved between human and mouse genomes, can you predict whether a given exon, a given part will be alternatively spliced or not?
mhm.
And
and
even that a subclass. But yeah, so
mhm. And what's the practical implication of that research? Okay, so we have
yeah.
human genome mice,
Hmm.
and then we see that okay, some are spliced, some are not. But what are the like? How can we use the results of this?
Yeah, so the first thing is, of course, at the fundamental biology level, you want to understand how alternative splicing takes place. And so, if we can develop good models of predicting whether there will be alternative splicing or not at a given exon intron boundary, then that can help deepen our understanding of the process. And the second thing is that if you think in terms of synthetic biology, then maybe it can also help design future experiments where the splicing is controlled through external factors. But that is something I've not really thought about much. So that is just something I made up
Mhm.
on the spot, really, the second thing. And the prediction was done using Bayesian networks because that was something that my lab had been using already. And then for comparison, I use support vector machines. And so one of the interesting things that I learned in my first project was that the features usually seem to be more important in determining performance than the algorithm, given that they were both strong models. And having said that, experts in any one given model can usually squeeze some extra performance out of that. But as a beginner, to me, there was not a big difference between the Bayesian network and SVM as long as we had the same set of features.
Mhm. Mhm.
And yeah, you were saying
Yeah,
something?
yeah. I mean, this is everyone who deals with machine learning at some point of time learn this that feature
Yes,
engineering
yes.
comes first, and
Uh-huh.
then the model comes second. And on Kaggle, it might be important, right? So the way you tune XGBoost, maybe people who are in the top they tune XGBoost a little better than people who
Yeah.
are, but there is always in the top participants, there is always somebody who just has a very smart idea, comes
Mm-hmm.
up with a super genius feature, and
Yeah.
then the model is simple, right? Instead
Yes.
of having layer upon layer upon layer of XGBoost models, they just come up with a good feature that just beats everyone else, right?
Yeah.
And
Yeah.
so, but in terms of features for your particular problem, what kind of features are there? So you have this genome data, right?
Yeah.
Uh, so which are strings of characters, if I
Uh-huh.
remember correctly.
Yes, yes, yes. They are indeed strings of characters, and then these boundaries where the splicing happens, they have specific motifs or character combinations, if you will, right? So it's like there you will have an AT on one side and a GC on the other side, and so on. So like there are some well-defined sequences around the boundaries. And then one of the features really is just that by looking at what exactly is the context around the boundary, you see that some motifs are predictive of alternative splicing. The probability goes up if you have these motifs versus those. And since it was for events conserved between human and mouse, the extent of sequence conservation around that region also mattered. So the more highly conserved the overall region was, the more likely that the splicing or lack of it would also be the same in two. And then there are features we derived which are to do with the biology of it. There is something upstream which plays a role. So we measured the strength of that sequence in a given sense and so on. So most of the features then become a little biologically involved. So maybe not so interesting for most listeners.
Mhm.
But two interesting things that I learned towards the end of the project, once we got reasonably high performing model or highly accurate model going with a balanced accuracy of maybe more than 95%, if I remember correctly. So then I was interested in knowing two things: that what are the things that my model is very confident about and yet are wrong? So the false positives with a high probability. And the other thing was the false negatives, that why are some things positives, but my model cannot identify them, even though it's overall quite accurate. And so the interesting thing was that when I looked at the ones which were false positives, they seemed to have everything in place which would make them alternatively spliced, yet the data said they were not. So what I did was that I looked for updated evidence, because the annotation was all based on some experiments that had been done, but it was now a few years since they had been done. So I looked at the databases to see what the current evidence was, and we could actually find that some of them were indeed positive if you used updated evidence. So
Mhm.
the model was not really even wrong, but it was wrong because the data was kind of outdated.
Okay.
So
So
that was,
it was, I think it's called label noise in machine learning.
mm-hmm,
So
yeah,
when you, even though maybe you have some label noise, but overall, if your data is good, that some, in some cases, you might have mistakes of labeling, but
mm-hmm,
this is fine. And then if your model is good, you can actually then later use this model to correct the original data. That's
yeah,
super interesting.
yeah, that was very interesting. And the other thing was that the false negatives. So I then isolated a few false negatives and tried to see if I could see something else which was common between them. And indeed, they turned out to have something else going on, which was conserved secondary structure, which was not something we had considered in our model at all. And so we found that by looking at the false negatives, we could potentially find a new mechanism. And but unfortunately, we were scooped by someone else. They published the same finding before
Mm,
us.
okay. So in science, if somebody publishes the same finding, what do you do?
I mean, usually nothing much. It's just bad luck and move on. But yeah, sometimes if you really want to publish it anyway, you could try maybe a very low impact avenue. But usually it's like the novelty is not there anymore, right?
Mm,
So
okay.
if you're very lucky, you publish right at the same time, or if you're
Hmm.
very big, then you coordinate it together, right? Like the human
Okay.
genome. So
Okay.
where the public and private efforts both published in the same issue of Nature.
Mm,
Sometimes that happens. Very big labs, because the editors know, so they say, "Hey, we know that they are doing the same thing, and are you okay with publishing together?" So then
mhm.
they kind of shake hands and publish different papers in the same issue.
Okay. Okay. So
But
in your
usually it's bad
case,
luck.
yeah, in your case, you just didn't know somebody was working on that.
Yeah, and the funny thing is they ended up being a visiting researcher at our lab the next year.
Okay. Well,
And
eventually
yeah.
you found out that you were working at the same thing and you started to collaborate, right?
No, I think it happened more because they wanted to spend a year in Germany. They were originally
Uh,
from Germany and then in teaching in California, and they wanted
mm,
to take a sabbatical in Germany.
okay.
And they found related work, not necessarily through that paper, but just, yeah,
Mhm.
overlapping
Okay.
overall
Okay.
area.
And is it related to the cancer research and like
So,
tumor modeling?
no.
No?
Well, so there is alternative splicing going on in cancer, but during my PhD, I did not work on cancer research at all. I worked just on the problem of alternative splicing and then different kinds of alternative splicing and in different organisms and so on. But I had always wanted to work in disease research. So
Mm,
when I was applying for my postdoc, I initially started applying for alternative splicing positions. And then I realized that if I don't move to cancer research now, maybe I'll never move. I'll just get more and more into the same kind of research. So then I pivoted and I was
mhm,
fortunate to get this position in Chris Sanders' lab at Sloan Kettering.
mhm.
So that's
So,
how I started in cancer
mhm.
research.
So how do people deal with cancer and tumors right now? What's the work you're doing?
Okay, so I mean, there are many ways of course, attacking cancer, and cancer is also very difficult to control, of course.
What is
But
cancer? Maybe we can start with that because
yeah, yeah.
I have very limited idea. I know that some cells they go wild,
Mm-hmm.
right? And then they start attacking other cells. So the other cells, it's like zombies, right? This zombie
Yeah.
cells. They turn and
Yes,
then it grows. Is
yes,
it
you
correct
could say
understanding?
that. Yes, so, and people say that you could say that cancer is not one but thousands of diseases because there are many different kinds of cancer with their own specific properties. But like you said, the one overarching property that is shared by all cancers is uncontrolled growth.
Mhm.
So, and there are again many theories of how cancer comes about, but the one that is most prevalent nowadays is the mutational theory. So, the idea is that our cells have many different processes supporting both growth and regulating growth or even forcing death of the cell, right? And what happens in cancer cells is that they hijack the growth processes, driving continuous growth, and they also disable the kind of watchdogs, right? So the processes which have to ensure that cell death or apoptosis happens when it needs to. So a cancer cell kind of tricks the body into letting it grow
Mhm.
infinitely. And so, for example, with the immune system, the cancer cells actually manage to hoodwink the immune system because of some changes they introduce, and the immune system does not think of them as foreign. If
Mhm.
it identified them as a threat, then it would try to eliminate them. And that's what immunotherapy nowadays uses, that it tries to restore, in a way, the recognition of cancer cells as foreign objects.
Mhm.
And yeah, so there are so many of the therapies are so the old therapies are more process related, like chemotherapy.
Mhm.
A
And
lot of
therapy is
yeah.
so the cells are growing, and we need
Mm-hmm.
to do something with this growth. We need to stop it, right?
Yes.
And
Yeah.
this is what the therapies are doing, right?
Yes. So the ideal case is where you really just eliminate the cancer cells altogether, but that's very difficult. So that's the ideal case. But also, if you slow down growth, then that can be like partial success, right? And some people think that maybe the best you can do is to turn it into a chronic disease. So it's not really completely gone, but it's, say, manageable. Even that is a difficult goal to reach. But so what I was saying was that in chemotherapy, in the kinds of therapy that we've had for several decades now, most of them kind of target fast growing cells in general. So while they do kill cancer cells, they also impact other fast growing cells like hair and nails, which is why many people who are undergoing chemotherapy may lose all their hair while they're being treated. And then when the treatment stops, their hair will come back. But that's a very common side effect and very striking one.
I had no idea that that's the reason. I know this is a side effect, but like
Yeah.
that the idea is to kill fast growing cells, and
Yes.
nails and hair are fast growing. I had
Yeah. So when the therapy is not specific enough to only target cancer cells, then all the other cells with same properties also get affected, right? So another class of therapy is anti-angiogenesis, which tries to slow down the growth of new blood vessels, which cancer cells need to grow. So that again, it slows down cancer cells, but it also, if you have a wound while being treated with those therapies, your wound will not heal easily because your wound also heals with the growth of new blood vessels. So it's like every kind of therapy can also have a specific kind of side effect unless it's extremely specific in targeting only cancer cells.
no. But
Uh,
we as humans, do we have some understanding why this happens in the first place? So I know, for example, my grandfather, unfortunately, he passed away because of lung cancer. He was
mhm.
a smoker, and while it's sad, everyone's like, okay, he was smoking, so that led to the disease. But there are
Mhm.
also many people who smoke and nothing happens, right? So they're
Yes,
healthy.
yes.
So like, why some people who smoke, they are more prone to that, while others like, is there an understanding how exactly this happens
Yeah,
in
yeah, yeah.
humans?
There is understanding and there's also gaps in understanding. So, for example, there are, like you said, there are smokers who don't get lung cancer, but also, unfortunately, there are non-smokers who get lung cancer.
Yeah,
So that's a
sure.
separate area of research that what's going on with these people who are getting it without smoking. Some of it may be explained by like secondhand smoking or working in smoke-filled conditions and so on. and others it's not very clear. And about people who smoke and still don't get lung cancer, you can have so-called protective mutations. So, which is something you see even outside of disease, right? Like some people eat a lot and still remain skinny just
Hmm.
because they have something which makes their metabolism different from others. And others may say that I am not even overeating, but I seem to be gaining weight all the time. And so there are many things where just a small difference of mutation can mean that your body reacts very differently. But in
Hmm.
terms of how it comes about, it's mostly seen as either being inherited. So if you're unlucky and your family just has one mutation that runs through generations, and that helps cancer, or it's through the accumulation of mutations throughout your life. That's why the non-inherited cancers usually arise later in life because you need enough time, and it's still a random process because so we have like millions, billions of cells, and there are new cells being produced all the time. And in all the copying, there are some errors and very few errors, one in millions. But it's just that there's so much happening that you will end up with some errors anyway, even with a very, very high fidelity copying mechanism. And then the body has some processes to repair these as well. But inevitably, over the years, you will accumulate some mutations. And
Hmm.
most of these mutations are benign, they don't harm you in any way. But if you're unlucky, you may get something that helps cancer by either promoting growth or hampering the regulation of growth.
Hmm.
And so, if you get enough mutations, and that may be just a small number needed depending on the cancer type and your predisposition, and then if you end up in this situation where you have uncontrolled growth, that's when you have cancer.
Hmm. And is this what you, because one of the questions we have here in the list is about your work on tumor modeling. Is
Hmm.
this what you do when you model tumors? Like you
Yeah,
apply these
no.
random mutations and then see what happens, or what is this work about?
Not me. I mean, there are people who do that. So there are different ways of doing computational biology. Some of it is highly theoretical, and some of that is really just deriving a lot of equations with a model of a cell and so on. And there are people who do that kind of research. What we did was is cancer genomics, which is really just studying the genome in different ways. And the tumor modeling there actually is not tumor modeling, but working with tumor models, which is a slightly different thing. So there are various kind of models that scientists have developed in order to study cancer in the laboratory. And the most popular and long-lasting models are called cell lines. So that's really you isolate some cells from the tumor and then try to grow them in the lab. And most of such efforts tend to fail because the cells are not really meant to be outside. And
Hmm.
so it's difficult to make them grow in a lab. And then people tried many things. And what ended up being the most successful is when you take cells from highly aggressive or metastatic cancers, they have a higher probability of lasting outside, given that you feed them the right kind of things. And but because of that, there is a bias in what kind of models you end up with. So then the more easy to treat cancers or the more or the less aggressive cancers often don't have good models.
Hmm.
The best models are all really of highly aggressive and metastatic cancer. So that kind of biases research in a way. So what we did was we took cell lines from a major resource, the Cancer Cell Line Encyclopedia, and tumors from another big resource called the Cancer Genome Atlas, and asked if the cell lines, to start with in ovarian cancer, really resembled the tumors. Because if they are good models, you would expect them to sort of have the same kind of features, similar mutations, and so on. And what we ended up finding was that some of the most highly cited, widely used ovarian cancer cell lines did not really mimic the tumor characteristics very well. So, and then there were some which were not used much, which were probably better models. So, and then we did follow-up studies and using different approaches and in different cancer types, etc. But the crux of it was really that these models of tumors, which are used by researchers all over the world, how well do they actually resemble the tumors in a genomic sense.
And genomic sense here, like how actually cells multiply, right?
So no, so it's actually looking at the common mutations, the mutations which are believed to be important in the cancer,
Mhm,
or at gene expression, copy number, and other things. So like mutations are the most common mechanism, arguably, but there are other ways in which cancer cells can shift the balance to their advantage. One of them is so-called copy number aberrations, because we get one copy of each gene from our parents. So by default, we have two copies of each gene. It kind of also protects us because if there is some problem with one copy, maybe the other one is enough to supply the needed functions. And what happens in cancer is that you can have extra copies of some regions of the genome and the genes contained in them. And sometimes you can have loss of some things. And so this is like copy number gain or loss. And as you might imagine, cancer benefits when the gain is of growth promoting genes and the loss is of the protective genes, which would
mhm,
ensure that you don't have abnormal growth.
mhm,
And so we also study copy number aberrations. And then there is gene expression, which again sort of quantifies how much of a given protein is going to be made from a given gene. And so variations again, you have high expression of growth promoting genes or low expression of protective genes that are called tumor suppressors. So the growth promoting genes are called oncogenes, and the growth suppressing ones are called tumor suppressors. And again, cancer always benefits from too much growth and not enough
mhm,
suppression.
mhm,
So we kind of studied data from which was collected from experiments related to all of these different ways in which a cancer cell can grow, and put that together. And so it's multimodal or multi-omics analysis when you study different of these omics data types and try to put the picture together.
mhm. And yeah, only yesterday I learned that there's this omics. So I knew genomics, but it turned
Hmm.
out that there are other omics as.
Yeah, yeah. There are transcriptomics and proteomics and many different kinds of omics. But yeah, all of that just kind of says that we are studying this in a large scale manner.
And how do you use machine learning for these things?
So, like what I was talking about earlier, so even that you can use machine learning for comparing tumors and cell lines. But so, and it also depends on what we consider machine learning, right? So there are some things like, say, PCA or
Mhm,
basic forms of clustering or linear regression, which many people consider these to be part of machine learning, and others would claim that no, these were already part of statistics or other
mhm.
fields, and machine learning in
Well,
their
let's say
view
any
is, yeah.
supervised or unsupervised
Mhm.
learning. So
Yeah,
linear regression to me is supervised learning,
yeah.
right? So you have some data and you want to predict a certain thing. So you have this prediction problem you're trying to solve, hence machine learning.
Yeah, so to give a very common example of clustering being used in cancer research. So, like I said, cancer is thousands of diseases. And then, even if you focus on a given organ, like let's say having breast cancer or lung cancer or prostate cancer, it's clear that not all those tumors are the same. There is variation even there. And so if you cluster, for example, using gene expression data or protein data, you can see that usually for most cancers, you can see distinct clusters, not necessarily a huge number. And it also depends on how you do the analysis. And we know that in clustering also, sometimes you can have different number of clusters from the same data, depending on the algorithm you use, the cutoffs you use, and so on. But so a famous example is that in breast cancer, there are believed to be five biologically distinct groups. They have different properties, different genomic alterations, and they do actually respond to different therapies. And so when you get a breast cancer data set by running the clustering, you can sort of identify that here are these five groups. This is the one that has the least aggressive cancer. They're likely to have better outcomes. These are the three which have these different sets of alterations, and accordingly, these different therapies are likely to work well for them. And here is this fifth group, which actually lacks all of these alterations and is the most aggressive type. And this people in this may be unfortunate and have the worst outcomes.
Hmm.
And that is driven by clustering and is now part of the standard way of analyzing breast cancer tumors.
Hmm.
And then you can do that. And people do that in some cancers, it's now established and part of the research. And in others, people do it in a prospective sense, right? Like, okay, so far we have been just treating this as one disease, but we can see that at the genomic level, this is n different clusters. And let's dig into that to see if we can deepen our understanding of this particular cancer and help come up with new ways of treating it.
Hmm.
So that is one common way.
Hmm. Yeah,
And
what I saw on Kaggle, I think there was more than one competition there where the goal was you have an image of a tumor
mhm.
and the goal was to predict if it's a bad tumor or a good tumor, right? How
Yes,
you
so
define, I don't remember the uh, how you call them, but like that I remember having these competitions on Kaggle. Uh, right? Did you take part in any of them?
No, but in my ML Zoom Camp projects, I did do one such project. So when I started on the projects, I really wanted to do something related to cancer research and also do an interesting project. And the first one had to be somewhat simple. So that was just the Wisconsin breast cancer data set, which is a simple tabular data set. And the second project, by that time, we had started the deep learning portion of the course. So there I used one of these data sets where you have these images and you have to predict whether it's likely to be a tumor or not. And
Hmm.
so I did that there. And you're right, that is something that has become much more popular in the last 10 to 15 years. Before that, we didn't have, I think, deep learning to do these things. You could have approached it to some extent, of course, but deep learning really brought about a revolution, right, in working with images and audio and video. And so in the last decade or so, there have been many papers which kind of even compare the performance of machine learning to pathologists.
Hmm.
And sometimes people upset pathologists by saying that machine learning was better than them and so on.
Hmm.
But I think the more agreeable view is that machine learning can help a pathologist do even better than they do on their own.
Hmm.
So, yeah, so that is definitely an active area, applying
Is it
deep learning
a
to.
part of standard checks these days? Like you go to a lab, you give them a sample, and then they run machine learning on top of these things, or some models, or it's still maybe getting adoption?
I think it is still getting adoption, and I think it's dependent on the institute whether a large number of samples or most samples end up in front of the machine learning people or not.
Mm.
Because I also read recently, and I didn't read the article yet, so I can't know in details, but there were a lot of startups which started a few years ago based around this idea that people will use machine learning to do the job of radiologists more and more. And then apparently most of those startups have not done very well.
I
So
assume
I
it's a
think, yeah.
very highly regulated area, right? It's maybe it's not the technology that is blocking the progress, but the regulations, and probably they're there for a good reason, right? I don't know.
Yeah, I think in areas like medicine and law, etc., that will always be there, even as AI gets more and more powerful. But when somebody has to take the blame, who's stepping forward? And I
Yeah.
don't think AI companies will step forward. They will mostly
Hmm.
want to have that as part of the contract that we are not responsible for the outcomes. You are
Hmm.
using it and use it to your best judgment.
Hmm.
But if something goes wrong, it's still your problem. So it's
Hmm.
still the doctors and hospitals or the researchers or the company which will kind of have to
Hmm.
be responsible, I think.
But I guess right now, if I go do a blood sample to check that, then it's my doctor who is giving me. So I don't know what he is doing. I recently, well, recently, last year, did a blood sample to get checked for prostate cancer. So in Germany, I think people from starting from 13.5.
Mhm.
can do this. Anyways, like it was just my doctor. He said, "Yeah, if I find something, I'll give you a call." He never called me.
Mm.
So it's a doctor who gets results from some lab, who looks at the data and then makes the call or doesn't make a call, right?
Yeah, yeah,
So
yeah,
then
but
at the
yeah,
end,
no,
it's the decisions, doctor's decision.
y- yeah, but there's also like a lot of data leakage along the process because it's like on the one hand, we have laws protecting the privacy of patients. Like here we have that called HIPAA. And when you work in a hospital in the research division, then you see that that's very tightly controlled and you cannot access data without signing a lot of papers, etc., etc. On the other hand, nowadays, apparently, hospital staff is using things like ChatGPT
Hmm.
to understand cases. And that
Hmm.
is where a lot of data is getting leaked to OpenAI and other companies, right? And
Yeah.
so that is potentially an unintentional violation of patient rights.
Hmm.
And that's something that with AI we will have more and more, that there's a lot of gray areas around your rights as a citizen, as a patient, and so on. But anyway,
Tell us more about the projects you did for Zoom
yeah.
Camp.
Yeah.
You said there was one tabular Wisconsin breast cancer data set, and the other
Mm-hmm.
was images, right?
Yeah, the other was images, I think, of brain cancer to predict whether there were tumors or not. And then by that point, it was like, okay, you have your two projects, you've passed them, so you don't need to do a third project. But do you want to be high on the leaderboard? And so
You
I
wanted,
was,
right?
yeah, I wanted that. So I decided to do a third project, but then I said, okay, maybe I should do something different this time. And so I did the third project on traffic sign recognition. So the German traffic sign data set. So that was an interesting and different project related to deep learning and
Mm,
images. And then I even did the competition and the article. So I left
okay.
nothing really.
Yeah.
So yeah, I think very
Uh,
few people did the article.
what was competition about? Which year did you take it? Was
So
it
it was last year.
demand
Yeah, right.
forecasting, right?
And no, I think it was before that. So it was some, yeah, right now it's evading me what what the competition was about. But I couldn't give much time to it, but I joined anyway to at least get
Yeah.
some points, make the
Okay.
benchmark for the points.
Yeah.
But yeah, but I was also trying to write the article at the same time. The competition and the article kind of were running into each other.
Hmm.
So it was like then I decided, okay, I will do the article and gave less time to the competition. But yeah, and then I did the LLM Zoom camp last year and the machine learning engineering Zoom camp just now.
Hmm. You mean MLOps, right?
Hmm?
MLOps one, right?
Yeah, the MLOps was just now. Yeah. So the
Yeah.
Zoom
Okay.
camp with LLM was last year.
Hmm.
And I meant to ask, there was going to be an open source LLM Zoom camp, I thought, or is that cancelled?
Yeah, it's kind of like there is a dependency on a sponsor. So they
Hmm.
promised to give us infrastructure so
Hmm.
everyone
Hmm.
can just train all these models on GPUs. But there is some bureaucratic problems.
Okay.
So it's a
Okay.
big company. So I hope it's going to happen, but they turn to be way slower than I expected. So
Hmm.
unfortunately, right now it's on hold until further announcements. But like once it's done, everyone will know about this. Like we will announce it everywhere. But for now, yeah, I'm just waiting. Since summer, actually, it's been half a year, so
Hmm.
it will soon be half a year. Yeah, but it is what it is. I'm sorry about that. I also want to do this course, but yeah, I'm just waiting. Anyways, what I wanted to ask you, so with these projects that you did around cancer, very useful for your work and not maybe not necessarily projects, but like this, what you learned in our courses regarding about machine learning, like how did you use this at work?
So I didn't really use that for work so far,
Okay.
but it helped me get into some parts. Like I hadn't really done deep learning. Like I mentioned, I had done some machine learning off and on for a long time during my PhD. Then I took Andrew Ng's courses on Coursera when they arrived the first time. So I had some background in machine learning. but then all my applications were for specific projects and publications in academia, and I had only done what I needed for a given project. So it was like, on the one hand, I did some parts of machine learning I had not touched before, and more than that, it was that I got into deep learning and also the work related to reproducibility. So I knew of Docker, but I'd not used it as much. And of course, I'd never used things like, say, Flask. And using different virtual environments also, I'd done a little bit of that, but it became more of a habit. So all those things were what I picked up from that, from the first machine learning Zoom camp. And then for LLM Zoom camp and machine learning engineering, those were more or less entirely new things for me because I had not worked on those things at all before.
Yeah, that's
Right.
interesting that you found these things interesting. I'm really glad to hear that. Thank you. Uh, well, maybe, who knows, maybe at your next job you'll get to use some of these things.
Yeah, I'm even considering potentially pivoting more to this area if needed. So it just seems like there are jobs, more jobs in AI and ML. And within my industry, it's like you have some jobs which require AI and ML together, or maybe even shifting areas if need be. So we have to see.
Mhm.
But right now, I'm just starting out with freelancing by listing myself on Upwork. And I see that on your channel, several people have appeared who've talked about freelancing as data scientist or machine learning experts and so on. And
Yes.
so, yeah, so maybe I will look a little more into that as well for the
Okay.
time being. Yeah.
Hope it goes well. Yeah, we
Thank
recently
you.
had episodes about that, but also there were episodes like year ago, 2 years ago. I think it's a recurring theme,
Hmm.
and people are quite interested about that. I did a bit of freelancing, but it was like 12 years ago or even more. Yeah,
Yes.
it's an interesting thing. So I was studying, and I had a bit of free time, and I thought, okay, like I can put this free time into some use.
Hmm.
So for me, it was not only extra experience, but also an extra source of income while I was studying. And then when I finished my master's, I had things to talk about to my future potential
Hmm.
employers, right? So that was very useful. But outside of that, I didn't really freelance much. But, yeah, so I would be curious to try this at some point too. I'd find it projects and then consulting. That's cool. Um, do you like? I'm still interested in like the main topic of this
Hmm.
conversation, the cancer
Yeah.
research.
Hmm.
Um, in your last position, like you've worked with this topic for quite some time.
Hmm.
What was the last thing you did like that is about cancer research?
So in the company where I was working before it was shut
It
down.
was the industry, right? It was not
Yeah, yeah, yeah.
not
Yeah,
university.
so it was industry, and so it was quite different from academia in the sense that there was a much larger team kind of working together. Because in academia, it was always like, okay, we are the computational people, and then you are the lab people, and then let's get together and do something. But in the company, it was like it was already that we are all part of one larger team pulling towards a different goal, not necessarily a publication, but a new drug. And
Uh-huh.
so it
So
was
it was
like
a
you,
pharmaceutical
so,
company.
and so, right, yes, and right from it was the research division of a pharmaceutical company. So the idea was always to find new potential drugs. And for that, the computational biologists and the molecular biologists and the chemists, they all work together. And so there it was one distinct feature was this, that you had a much larger sense of a team. And then as the computational people, we started out by identifying potential new molecules which could be used to develop a new drug. And for that, we again studied many different kinds of omics data. And I also found out that when you're in the industry and you have a lot of money, then you can get extra data. So it also changes the kind of research you can do if you have the bandwidth to do research. So the same resources from the same institutes, there's a public version and then there's sort of a premium version. And so in academia, I had always worked with the public versions. And then in the company, I saw that, okay, it's the same research institute, the same database, but then you have this, you kind of are half a year ahead, or depending on how the source institute does it, they can give you a lead of maybe half a year to 1 year over n- everybody who's not paying them.
Interesting. I'm just curious, how does a day in a day of somebody who is working in drug discovery industry, how does it look like? Did you go like, I don't know, was it just office work in front of a computer, or you worked also in a lab, or like how did your day look like?
So I never worked in a lab. I mean, there are some people maybe who have more of a background in biology and then move towards computational biology. Maybe they might, but usually it's more specialized. So we were only doing computer work, but this was post-pandemic, so it was hybrid. So you don't go into the company every day anyway. Half of the time you're working from home, but maybe you coordinate with your colleagues when you go in. So you have a weekly meeting of the computational people, and then many of us would try to definitely make it to the office on that day so that we could also meet in person. And then you go on some days coordinating with your collaborators from other departments because they may not be coming on that day when you have your colleagues coming in. So part of it was driven by just the people that you need to meet in person.
Mhm.
When would they come and when would you come? And so
But
maybe,
I'm also wondering about like
and
the specific work. Um, is it like, I don't know, you come and you open your computer and you work in Excel or some sort of
Mhm.
like some other environments where
Yeah,
you want to do data analysis, or what was
yeah,
the
yeah,
job
yeah. Yeah, so
like?
I mean, if you're working in Excel only, then you're probably not a computational biologist, right? So then you're
Yeah,
probably
I
just
have
a
no idea.
biologist.
Like I
Yeah.
don't
Yeah.
like
No, so if you're a computational biologist, you're definitely doing some programming.
like R, I guess,
And
right?
yeah, it could be R, it could be Python. Some people do other things. And also, if you're doing like statistical analysis, then you could use other packages also, right? Like maybe you use SAS, maybe you
Mhm.
use SPSS. But in academia, especially, most computational biologists would tend to work with R because it's free and it has all the routines that those other SAS and SPSS would have. And you don't need necessarily like a GUI, right? If you are a programmer, then you can
Mhm.
program from the command line and Vim and so on. So within editor and so on. So yeah, and R is extremely popular. Python is also used for quite a few things. And of course, as you know, if it's machine learning and especially deep learning, then Python is usually going to be more convenient than R.
Mhm.
R kind of takes the Python deep learning packages and runs them in R. So for
Yeah.
machine learning, it can be R, but for deep learning, it's really like either it's Python or it's like Python pretending to be R. So,
Yeah, yeah.
but
Okay.
yeah, so yeah,
Uh,
it's and a lot of meetings and reading papers. So you
okay.
have to read the latest research to be on top of things.
Mhm.
So it's a mix of that
Mhm.
coding, thinking, and collaborative discussions and reading
Mhm.
research. And if
Yeah.
you're lucky, you may do some research of your own.
Okay. So I guess the main difference between, let's say, me, I was working in an internet company. So for me, the goal was, let's say, increase conversion of a certain thing, right? But for you, it was kind of similar. So the work was still programming,
Mm-hmm.
but the goal was to, let's say, develop a new drug, right? And like some things, some analysis that you needed to do in order to, I don't know, find out how to do this, right?
Yeah, I think you can say at an abstract level is really the difference of the domain, right? So
Yeah.
the computational bits remain similar. So you're still doing supervised learning, unsupervised learning, and so on and so forth. But and there's a lot of data cleaning and quality checks also required
Hmm.
because if you're not working with the right data, then everything else is meaningless, right?
Hmm. Yeah.
So
That's
it's
true everywhere.
yeah, and we have a lot of things which were underappreciated until recently. So in biology, you can have batch effects. So
Mhm.
experiments run on different days can have similarity just because of the day they were run on. And so there were papers where there were findings that people later found out were wrong, because they
Hmm.
had not taken the batch into account. So now it's like a standard part of your analysis that you correct for batch effects before you do downstream analysis. But
Hmm.
for a while, people did not take the batch into account, and so there were papers published where some of the findings were really just driven by when the experiment was done and not by real biology.
Hmm.
So there's a lot of such things, checks that you have to do as well if you're
Hmm.
doing a proper job. But
Yeah,
yeah,
interesting.
otherwise it's similar at a certain
Hmm.
level.
Yeah, I had no idea. Like, usually it's not, let's say, in my bubble. Like, I don't
Hmm.
hang out with computational scientists
Yeah.
or biologists. Well, my sister was a biology, but then she dropped out of PhD
Hmm.
and she works in logistics right now.
Oh,
Yeah,
big
it's
change.
like research. Yeah. But yeah, I had no idea what exactly what she was doing. I know she was taking samples from lakes, but
Oh,
that's I
okay.
know.
Like
Yeah.
ecology or?
Yeah, I guess she was also working in Germany.
Hmm.
Maybe I should ask her what she was actually doing. Okay, maybe last one before we finish. You've
Hmm.
taken a lot of courses in Datadogs Club, I assume also outside. But regarding these Datadogs courses, because I assume students of this current ML Zoom Camp cohort might be watching you.
Hmm.
Any recommendations? Do you have any recommendations for them?
In terms of like materials or how to approach
Just in
things?
general, like I don't know, you've been through three courses.
Yeah.
Um, like I don't know, do you have any advice?
Yeah, so I think so personally I enjoyed all three courses, but I found ML Zoom Camp to be like the most organized one. And I think it's partly because when I took it, it had already been offered a few times. Whereas LLM Zoom Camp was A, offered for the first time, and
Yeah.
B is just an area that is completely crazy right now with these new releases every week. And so, how can you kind of have any stability, right? It's like everything you're learning is outdated in some sense the next week and so on. So LLM Zoom Camp in that sense was different, but also enjoyable. And then for machine learning, so MLOps Zoom Camp, it's now so mature that basically we followed recordings, right? So
Mhm.
it was again a different kind of experience. So I think ML Zoom Camp was a great experience, and if you're new to machine learning, I think it will also be quite hard to keep up. I was
Mhm.
fortunate in that the first part I had some familiarity with the machine learning part, so the really new stuff only started with deep learning. Before that, it was more like developing some new habits and so on. And but I really like the format you have for all your courses where you teach a new thing each week, you have homework, so it helps people refresh. And so I would, one advice would be to stay on top of the homework.
Mhm.
And yes, in theory, you don't need them for the certificate and you can just go straight to your project, pass your project and get a certificate. But the homeworks really help to reinforce your understanding of the concepts. And if at all you're interested in the leaderboard, of course, you can't afford to let go of any of
Mhm.
that.
Yeah, you were
And
pretty interested in that, right?
yeah, and it's also good to tweet or to post on social media your work, and that can also lead to unexpected things. I was resistant to it to begin with because I was very conscious of potentially adding noise to the feed of my friends and
Hmm.
colleagues. So the first few weeks I found it hard to post about my learning much because I was like, why would they be interested? And it's kind of like, so initially I was just driven
Hmm.
by almost unwillingly doing it, but then like I wanted the points as well. And so, and
Hmm.
then I got used to it and started doing it much more.
Hmm.
But I think it's like, it really depends on how you feel about
What
it.
did your friends
But
say about your posts?
so they mostly didn't say anything. So
Okay.
the comments that I got were actually from strangers who happened to maybe be following the hashtags machine
Uh-huh.
learning or deep
Okay.
learning or so, right? So I got
Yeah.
the occasional comment from that, or I got a few likes from the organizations because I tried to use include their handles, right?
Uh-huh.
So if it's like Docker, I would try to do add Docker, something like
Yeah.
that. And so sometimes my tweets were liked by those organizations. So
Okay.
yeah,
Okay.
but ultimately, I guess it ended up not mattering to my friends much.
Yeah.
But
Okay.
from friends and colleagues, I'm getting much more of a reaction on LinkedIn, where I
Yeah.
did not post my weekly
Yeah.
learnings, but I have posted about getting the certificates.
Mm.
and I have posted about this podcast, and there I get much more reaction from people that I actually know
Okay,
on
okay.
LinkedIn. Yeah.
Okay, so maybe to summarize, like if you're worried about the judgment of your friends or maybe polluting their feed, at the end they will not see or care anyways, right?
Probably, probably, and it may connect you to strangers, and
Yeah.
maybe it may lead to
But
some
that's
opportunity,
what
right?
you want,
So,
right? I mean,
yeah,
that's the
exactly.
idea behind learning in public, that it may lead to some connections that otherwise might not happen.
Yeah, and for the projects, I would say, so I often tended to try to max out everything and then ended up at the last stage scrambling to finish.
Hmm.
And so that's also with the homeworks. Often it was like I came to know that you kind of have a grace period because the deadline is midnight in Berlin, but you don't close it until you wake up or something. So you have a few hours. And so I spent many
You're
nights in the
telling too many
US.
secrets.
Yeah. So,
Okay.
so for the project, I would just advise them all
Mm.
to not wait till the last minute and then finalize everything, because if you have 2 weeks or 3 weeks, maybe get a lightweight version of your project working end to end first
Yeah, that's
before
very good
going
advice.
too much. So I think because that leads to a lot of problems where you're trying to perfect each stage and then you end up with something that is not working and you have half a day left and you're going crazy. So just get a lightweight version working and then try to do anything fancy if you want to after that.
Mm. Okay. Yeah, I realized that there are some questions that I sadly missed. Sorry about that.
Yeah.
But there are some comments like about premium data, for example. And there is a question if recording will be available later. Yes, it will be. Do you have time maybe to take one more question? Because
Yes, yes,
I feel
I
bad
have time. Yeah.
about leaving aside the questions we
Yeah.
have from the audience. Because
Yeah. No, no, I have as
for
much
the
time as you have.
activity,
Yeah. Yeah.
okay.
If you have time, I have time. Yeah.
So maybe let's keep it short, but still
Mhm.
answer this. What are the biggest challenges in working with cancer genomics data today?
The biggest challenges. I
Yeah.
so I don't know. I look at it more as a privilege than a challenge. So there is really no unsurmountable challenge other than for certain problems you may not have enough data. But if you have data, then every challenge is an opportunity, right?
Hmm.
So
Okay.
really, sometimes you don't have enough data.
Okay, so data is the challenge. And another question, which is actually quite related, question from Natalia. What kind of projects or data sets would you recommend for students who want hands-on experience in cancer bioinformatics?
Yeah, so fortunately, there are a lot of public resources now. So one is what I mentioned, the Cancer Genome Atlas, or abbreviated as TCGA, because that is the four letters in the DNA, right? So they try to be clever about it. So TCGA actually is a public project, and they can find data also hosted by the National Cancer Institute, NCI. And that project was specifically started to provide a gold standard collection of data. So across more than 20 cancer types, eventually more than 30 cancer types, I think. And for each cancer, you have hundreds of samples and many different kinds of omics data set. So they can really use that to work with any individual kind of omics or bring them together into multi-omics analysis. And so that's a really standard data set in cancer research. And then there's the Cancer Cell Line Encyclopedia, CCLE. And they can also find, I'm sure, review articles about this. And review articles are often a good place to find about all the major resources that you can use. And there are many tutorials, there are very good YouTube channels.
Mm.
And so there's a YouTube channel called Chatomics. So
Chai,
chat
chatomic,
and omics, Chatomics, yeah. It's a made
chatomics.
up word from, yeah. And that and many other channels, they have walkthrough tutorials that they can follow to
Hmm.
familiarize themselves with different methods of analysis and different data sets and so on.
Hmm.
And if you're a little experienced, then one very useful exercise is to try and reproduce a paper. Try and
Uh-huh.
reproduce all the results and images. You will learn a lot doing that.
Hmm. Yeah, I think it's true for any kind of domain, right? So in machine learning,
Yes, yeah, yeah.
somebody is making a report on something, trying to reproduce it is always good. Maybe
But I
another one, but
think machine
yeah,
learning is especially good for that because there's a
yeah,
culture of producing
yeah,
code, of sharing code,
yeah, yeah.
which is a little lacking in other domains, but it's getting better.
Okay, maybe let's take the last one.
Hmm.
Um, um, I assume a question from a listener from Germany. Did you
Hmm.
hear of projects in Germany doing this kind of research? And this was asked like 20 minutes ago when we were talking
Hmm.
about your work that you do
Hmm. Hmm.
in Boston, I guess. So,
Yeah.
tumor modeling and this kind of stuff. Do you know if there are any companies or institutes like research labs that are doing this kind of research in Germany?
Yeah, for sure. So I did not do such research while I was in Germany. I did mine all in the US. But of course, in Germany, there are many labs which are working in cancer genomics. And so if they search for that, they should easily find many. I think, for example, there are institutes in Berlin as well, which are working in this. I generally tend to remember more the name of the researchers than the institutes when I've not really actively
I
worked with them.
just Googled Cancer Research Berlin,
Mm-hmm.
and there is this very big company called Charité that
Mm,
has
yes. Yeah.
hospitals
Yeah. Yeah. Yeah.
in
Yeah. So
Berlin.
Sharit
So
would
they have
have.
cancer center.
I think there's a Max Planck Institute for Molecular Genetics. You have research labs doing this work there. There's DKFZ, the Cancer Research Center in Heidelberg.
Yeah,
So
I can
that is
see that
a,
they have
yeah.
also Center for Tumor Diseases in Berlin.
Mm-hmm.
And
Yeah.
DKFZ stands for
Yeah.
Deutsches
Yes.
Krebsforschung.
Zentrum, right?
Basically, like
Yeah.
it's a German
Cancer
cancer
Research
research. Exactly.
Center. Yeah. 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 So that's in Heidelberg. I had friends
But they
there
have an
at
office
the,
in
yeah.
Berlin. Like I
Uh-huh.
just Googled what I said, Berlin cancer research, and one of the one of the results is from this um, DKFZ that you mentioned. So they have an
Yeah, and if they also look at EBI, the European Bioinformatics Institute, that is based in Oxford, but it has partnering organizations throughout Europe and including in Germany, and they even have exchange programs between UK and Germany and so on.
okay. Yeah.
So yeah.
Thank you. Well, even though I accidentally forgot about the questions because I was so interested in talking to you about your experience, I apologize for that, but like we managed to cover them too. So thanks a lot, Rileen, for doing
Thank
that.
you so much.
But for joining us today, for answering the questions, for telling us more about cancer research. Like for me, this is a completely new area, so for me it was especially interesting because I had no idea about that. So thanks for doing this, and also thank you for being active in our community, taking our courses, and also giving tips for future learners.
And thank you so much for your great courses and your generosity. And I look forward to learning more with you, perhaps in the AI Dev Tools course.
Yeah, so that is happening. And
Yeah.
the open source LLM, I'm keeping my fingers crossed that it will work out. But for now, yeah, AI DevTools.
Yeah. All right.
Okay,
Thank you so
so
much.